Hi everyone. For chapter 25, we will discuss the different pathways of metabolism, nutrition, and energetics. If you remember some of the things about macromolecules breakdown and cellular respiration from your Bio 1406 lecture, then this chapter should be very straightforward for you. Now, metabolism is the sum of all chemical and physical changes in the body tissues, and that includes both catabolism, which is breakdown of large molecules into smaller ones, and anabolism, which is synthesis of large molecules from smaller ones, both of which we have already mentioned in the last chapter with digestive system. But back in that chapter, we were mainly talking about the mechanical breakdown and organs involved for the catabolism. In this chapter, we will go into the biochemical level of these processes. So these processes include how we are able to extract energy from the nutrients that we ingest, how we use that energy, and how we are able to store any excess energy if we end up not needing to use them all at once. All of these require the usage of our energy currency, which is ATP, which stands for adenosine triphosphate. For catabolism, we are breaking bonds between large molecules so we can extract ATP, mainly through the process of oxidative phosphorylation. I will talk about what that is later in the lecture. Or we can use catabolism to get our smaller building blocks like amino acids. Fats, for example, are broken down into their individual fatty acids and glycerol molecule through the process of lipolysis. Carbohydrates can be broken down into their individual glucose through either glycogenolysis or glycolysis, and proteins can be broken down into their individual amino acids through the process of proteolysis. Now, these three things we'll talk about as the macromolecules, the main macromolecules that we will ingest through our diet, and then we'll talk about how we are able to break these down into these individual monomers through chemical reactions. We'll talk about breakdown, and we'll also talk about the making of. So for anabolism, we're making bigger molecules from smaller individual monomers. The diagram here shows you or gives you a good hint of what is catabolism versus anabolism. This is going from a bigger molecule into smaller individual one, breaking these bonds right here, breaking all of them apart. So this is catabolism. Whereas here, you're building from individual little molecules and then connecting them with bonds. You are producing or making bonds. Hence, this is talking about anabolism, making of larger molecules. So anabolism is when we use ATP instead of spending ATP. Processes for structural repairs where we have metabolic turnovers is when we will use ATP. Remember those cells and enzymes we were talking about that needs to be regenerated within the digestive system or anywhere else in your body. Cell growth is also need the process of anabolism, such as mitosis or meiosis. Those cell divisions itself is where we need extra proteins and organelles. In places where we need to produce a lot of secretions like insulin, pepsin, stomach acid, all those things we already mentioned and talked about in the digestive system is also where we need to do a lot of anabolism. And lastly, ways we can bank or save our excess nutrient reserves, either in forms of fat through lipogenesis or in form of glycogen through glycogenesis, or lastly in the form of proteins through protein synthesis. And we'll talk about all these anabolism pathways when we get to that part in the lecture. Now, this is a good diagram that shows all those processes of catabolism and anabolism, and they all go hand in hand depending on different needs of the body in response to different environment. It's how our homeostasis of metabolism and energy supply is maintained. So this will serve as a good visual aid for you when you are studying for this chapter, because it summarizes the three types of macromolecules that we ingest through our diet, what are the processes that breaks it down, and what are the processes that makes those macromolecules. Make sure you can try to identify all of these processes as you're going through the lecture, or come back to this diagram after you're done studying this chapter, because this will be a very helpful diagram for you. Before we get into the catabolism or anabolism of different macromolecules, let's review what we have learned from our general biology classes on cellular respiration. Remember, cellular respiration mainly relies on a bunch of what's called redox reaction, where there is a reactant that is donating electrons or in form of hydrogens through oxidation reaction, while the reactant that are accepting electrons or gain of hydrogen is going through reduction reaction, both of which involve some transfer of potential energy between molecules so they can be used to power something else. So, for example, the electron transport chain, which is the last step of cellular respiration, relies on passing electron through a series of redox reaction to pump hydrogen ions that will ultimately be used to power your ATP synthase for production of ATP. The last part of the electron transport chain has oxygen as its final electron acceptor. So that's why throughout the entire process of electron transport chain, we are seeing redox reaction and happening between all those complexes, those complex proteins. So oxygen in the end was reduced to water through cellular respiration. Remember, the chemical reaction of cellular respiration, or the breakdown of glucose, is C6H12O6, which is glucose. With addition of 6 molecules of oxygen, you can get 6 molecules of CO2, carbon dioxide, and 6 molecules of water. Glucose here is doing the oxidation part, okay, being oxidized into carbon dioxide, oxidized, whereas your oxygen here is being reduced to water. Remember, it's easier to think about this process as gain of hydrogen or loss of hydrogen. If glucose has lost its hydrogen molecule, so that's why it is oxidized into carbon dioxide. Oxygen, on the other hand, has gained hydrogen, so that is why oxygen is reduced to water. So let me erase that so we can continue and not be bothered by that. Okay, so the process of cellular respiration involves coenzymes or electron carriers called NAD and FAD, which are, the long word for it known as nicotinamide adenine dinucleotide or flavin adenine dinucleotide, both of which are reduced to NADH and FADH2 when they accept electrons throughout the steps of cellular respiration that we will go over and review with you in a bit. So don't forget, these are electron carriers or coenzymes that will be responsible for moving electrons from different steps of the cellular respiration into the last electron transport chain to make ATP. So for cellular respiration, if you remember that we can make about 30 to 32 molecules of ATP from one molecule of glucose. The process of glucose metabolism is divided into 3 steps: glycolysis, Krebs cycle, and electron transport chain. The first step is called glycolysis. It is considered as anaerobic, which means it does not need the oxygen in order for the reaction to proceed, and it happens within the cytosol or the cytoplasm of the cell, not the mitochondria. Only when you have proceed into the Krebs cycle and electron transport chain does the cellular respiration happen in the mitochondria. This step, which is glycolysis, will break one molecule of glucose into 2 molecules of pyruvate or pyruvic acid, which then loses a hydrogen ion to make your pyruvate. It has a net gain of 2 molecules of ATP. If you look back into your notes, maybe from a previous biology textbook, okay, you will see that it actually makes 4 ATP, but because we actually need to spend 2 ATP molecules within glycolysis, that's why we make a net gain of 2 ATP molecules. Since red blood cells don't have a mitochondria or so much less any other organelles, as remember when we talked about the maturation process of the red blood cells, we mentioned that they kind of dumped off a lot of the unnecessary organelles within the red blood cells so that it is just mainly packed with a lot of hemoglobin proteins. But if they don't have mitochondria yet, they still need some ATP to power the cell's process. They actually rely primarily on glycolysis for making ATP. They can't do Krebs cycle or electron transport chain because they don't have a mitochondria. Skeletal muscles will rely on glycolysis only for energy during active contraction, and I'll explain what I mean by that in a bit when we get to fermentation. So, which is right here. So when oxygen supply is low, we cannot go on to Krebs cycle and electron transport chain because the last step of electron transport chain relies on having an oxygen molecule as the final electron acceptor. So when you are in anaerobic conditions, you don't have oxygen. When you don't have oxygen, your electron transport chain can stop running. When your electron transport chain stops running, all your NADHs will have nowhere to drop off electrons, because that's the whole point of electron transport chain, as we will discuss in a bit, is for your electron carriers to deliver and drop off their electrons. But if you have nowhere to drop their electrons, then you end up having all these NADHs with nowhere to go, and they're sort of just stuck there. The entire process of ATP production then is backed up when that happens. We need these electron carriers in order to go through the glycolysis itself. And hence, either as temporary means in human cells or in some bacterial cells, is what they do all the time, they will do a reaction called fermentation. Again, the purpose of fermentation is just to provide another means for NADH to drop off its electrons so it can be oxidized back into NAD+. So remember, here in glycolysis, you have NAD pick up electrons to make NADH. This is supposed to travel to electron transport chain to drop off electrons. Okay, chain. But if you don't have that electron transport chain due to absence of O2, then you need to somehow be able to drop off electrons somewhere else, or else you'll never get your NAD plus back to do more of this part of the glycolysis. Okay? All right, so fermentation will reduce your pyruvate into some form of lactic acid or lactate, okay, which will then also oxidize your NADH back to NAD plus, so you can go back here to do more glycolysis. Without this oxidizing step, you cannot do any more glycolysis. You have to free up your NAD plus in order to do more burning of your glucose molecule and turn that into ATP. If glycolysis stops, then no ATP can be made. But there is a byproduct here in form of lactic acid. For humans, they will travel to the liver to be neutralized once you have enough oxygen supply again, but buildup of lactic acid in muscles, however, will lead to fatigue, and that's why you can only rely on fermentation to keep running. You cannot rely on fermentation to keep running like a Superman. You can just continue making lactic acid forever, and you can keep running and running and running. You will have to eventually stop because the buildup of lactic acid will give you the sensation of muscle fatigue. So if oxygen supply is adequate, we can move on to the next step of cellular respiration involving aerobic metabolism, which means these are the steps that does require oxygen, and that happens within the mitochondria. The second step of cellular respiration is the Krebs cycle, or known as the citric acid cycle, or also known as the TCA cycle. There's a couple names for this process, but it's all the same thing. It happens again within the mitochondria and is considered to be an aerobic process, which means it will require oxygen to proceed. Notice that we are not using oxygen within the cycle itself. You don't see any O2 around, but remember how I talked about you need oxygen in order to oxidize that NADH, because oxygen is that final electron acceptor within the electron transport chain. If we don't oxidize NADH back to its empty electron carriers as NAD+, we cannot use them over and over again. And hence, without oxygen, the electron transport chain stops, and hence all the NADH will be backed up to even the Krebs cycle, and things will not proceed. You see how they're supposed to be dropping off electrons here, here, and here as well, where we are dropping electrons off to NADH. But if you have no NAD plus to begin with, then you can never have drop of electrons, and the entire Krebs cycle also stops. It's almost like the entire assembly line of a factory completely shuts down when you don't have oxygen. All right, so before the start of Krebs cycle, pyruvate from glycolysis actually needs to be modified through an intermediate step that convert pyruvate into acetyl-CoA. Okay, acetyl-CoA right here. So this is called the intermediate step. And it uses the enzyme called coenzyme A, and hence that's why it makes acetyl-CoA. This involves a chemical reaction called decarboxylation. Decarboxylation literally means the removal of a carbon dioxide molecule. That's why it's called decarboxylation, removal of carbon and oxygen in a sense. Now, you don't have to worry about all these intermediate products within the Krebs cycle. I won't ask any of those on the exam. You won't be tested on the names of the intermediates of the Krebs cycle. What you need to at least know is what the end products of this cycle are. Okay, if you go on to take a biochemistry class, then you probably need to know every one of those intermediate product and probably have to know how to draw them as well, but that's for biochemistry class. This is just an anatomy class. So what you need to at least know is what the end products are. So from one pyruvate molecule, okay, you will make 3 molecules of carbon dioxide as byproduct due to its removal from the reactants through decarboxylation. And hence, this explains why we have to breathe out carbon dioxide during respiration. In order for our cells to be alive, we have to keep making ATP, which means we have to break down glucose, which means we have to go through cellular respiration, which then means we need to breathe in oxygen or else everything stops, as I have discussed with you. And we need to breathe out carbon dioxide in return because we are making it as byproducts throughout the entire process of cellular respiration. So 4-NADH is the other thing that we are making through the process of Krebs cycle. We will be picking up electrons and hydrogens from different intermediates of the cycle, which then those electrons will be given to your NADH molecules here. So that's why we have 4 total gain of NADH. Now, 1-FADH2 is gained right here, FADH2. This is a special one, a different type of electron carrier that only picks up one pair, pick up electrons at the citric acid cycle. It doesn't pick up electrons from anywhere else. And in the end, you also have production of 1 ATP or GTP molecule. Now, because 1 glucose molecules mix 2 pyruvate molecules, all of these gains would be doubled if we're talking about 1 glucose molecule. Okay, so one molecule of glucose is equivalent to 2 molecules of pyruvate, which then through Krebs cycle is able to make 6 CO2, 8 NADH, 2 FADH2, and 2 ATP molecules. Okay? Now, it doesn't matter that GTP is made instead of ATP here because ultimately it can be used to make a molecule of ATP as well, though through what's called the substrate level phosphorylation. Phosphorylation usually indicates addition of a phosphate group to a molecule. So here's that phosphate group, and we're adding that phosphate group to GDP or ADP to make your triphosphate. So substrate level phosphorylation specifically means we are directly moving a phosphate group from one molecule to another molecule. This is different from oxidative phosphorylation, which happens within the electron transport chain. Okay, so in a sense, that's why I said it doesn't matter if we're making GTP or ATP, because whether it's GTP or ATP, we do have that phosphate group within the GTP that can be transferred to ADP, so that you will make ATP. We know how you see how one of the phosphate group from GTP was given to ADP, so you end up with adenosine triphosphate from the GTP molecule. The electron transport chain is embedded within the inner membrane of the mitochondria. Again, the mitochondria is one of the organelles within a cell that has a unique feature of having two layers of this phospholipid bilayer membrane. There are multiple sets of this electron transport chain within the mitochondrial membrane. So here you can see this is the mitochondria. Here is its outer phospholipid bilayer membrane, and these curvature here are your inner layer of the inner membrane of the mitochondria. So when you zoom in to this particular area, this is representing the outer membrane. This is representing the inner membrane of the mitochondria. This is different from the plasma membrane that borders the cell, which is not made up of two layers. And I'll explain why it is critical that the mitochondria has these two layers. So a set of electron transport chain include a series of these protein complexes, complex 1 through 4, very conveniently named. They are integrated into the phospholipid bilayer, which is the inner layer or inner membrane of the mitochondria. Their main job is to do a bunch of redox reactions, like I mentioned earlier, which will oxidize all those NADH and FADH2 that are made within glycolysis and Krebs cycle. Their other job is to pump hydrogen ions into this intermembrane space of the mitochondria, and also they will use oxygen right here as the final electron acceptor. Where then hydrogen will be combined with oxygen and it will be reduced to make water. And that's why water is a byproduct of cellular respiration. This process produces almost 90% of all ATP made in cells, where one glucose molecule can make up to 28 molecules of ATP just through electron transport chain alone, whereas glycolysis and Krebs cycle only makes about 4 ATP molecules through substrate level phosphorylation. Hence, this is why oxidative phosphorylation, which is what happens during electron transport chain, is what makes almost 90% of the ATP within a cell. Now, what do we mean by oxidative phosphorylation? It means that this is an indirect way of making ATP, where we are not moving phosphate groups from one by one from one molecule to another, but we're relying on a concept called chemiosmosis. Chemiosmosis is when these protein complexes right here are pumping out hydrogen ions from your mitochondrial matrix into the inner membrane space. The buildup of these hydrogen ions within the intermembrane space creates a concentration gradient that will allow for hydrogen ions then to flow back into the mitochondria via another integral protein called ATP synthase. ATP synthase. The ATP synthase is like a little turbine that turns every time a hydrogen ion flows through the molecule, turning it to generate ATP. This is why the mitochondria needs to have two membrane layers to be able to have a space or compartment that will allow for a buildup of these hydrogen ions within this area right there. It will ultimately power your ATP synthase to make ATP. If there was no hydrogen ions in this area, none of it will flow through the ATP synthase, nothing turns within the ATP synthase, and you cannot generate any ATP. And hence, this is why this intermembrane space is very critical and why the mitochondria has to have two layers of phospholipid bilayer. If you don't have this layer, let's say if this layer was completely eliminated, then these hydrogen ions will just float away to the cytoplasm, and you don't have a concentration gradient anymore of hydrogen ions. But we're relying on the hydrogen ion concentration gradient in order to power this ATP synthase. And it is easy to remember the name of this protein because it literally tells you it is for ATP synthesis, hence its name, ATP synthase. Now, with the process of electron transport chain, well, we've been talking about how we're able to make a lot of ATP, but exactly how many molecules are we able to make of ATP from an NADH or FADH2 electron carrier? Here it is. One NADH will make about 2.5 molecules of ATP, whereas one FADH2 molecule will make about 1.5 molecules of ATP. So you may need to know this for the exam. Be sure to make a note of this, sort of unit of how many unit of ATP you're making per molecule of NADH or FADH2. Hence, this is a summary slide of the three main steps of cellular respiration and how many ATPs are made in each step. From electron transport chain itself, which is right here, you are able to make 28 molecules of ATP. So you can see why it makes most of the ATP that's generated by your cell from one glucose molecule. Cellular respiration can generate about 32 molecules of ATP. This number may vary depending on which textbook you're reading from, and even here it says 30 to 32. Some place you'll see it saying it makes 38 ATP. Somewhere it will say 36. For exam purposes, you should at least know it is in the realms of somewhere around the 30s. That should be correct in terms of ATP. The exact number of ATPs is not black and white, depending whether you account for some of the electron losses as different molecules are traveling in and out of the mitochondrial membrane. And hence, we don't want to get to that complicated. So somewhere in the realm of 30s is correct. It shouldn't be in the 40s, 50s, or 60s, but the general number of 32 ATP should be pretty correct. Okay. So that was the whole process of glucose catabolism that you have probably heard of many times in the past biology classes. What about in the case where we need to make glucose? Now, remember that in 1406 biology class, you learned that plants are able to make glucose or synthesize glucose through photosynthesis because they have chloroplast within their cells, and within chloroplast they have chlorophyll. That's why they look green. Now you look at your skin, we don't look very green because we are not plants, and hence we can't do photosynthesis to generate glucose molecule just by nicely standing outside in the sun. That would be nice though. We don't ever have to eat again if we can do that. But because we cannot do photosynthesis, we have other means of generating glucose in case where our body runs out of glucose molecules for glycolysis. Now, so what we can do is call gluconeogenesis. It is done by our liver cells. Remember from last chapter, we talked about how the liver has a lot of metabolic regulations for our body, including carbohydrate metabolism. Liver cells can make glucose from non-carbohydrate molecules. You don't need to know the details of the mechanisms and what exactly those reactants are. Just know that somewhere along the way of glycolysis right here, it can add reactants molecules that will remake those intermediates of glycolysis at different points, and that will ultimately be able to produce molecules of glucose within the liver. So that's why going this way is the process of gluconeogenesis. Remember, though, that gluconeogenesis is not a reverse process of glycolysis. You have different starting material. Just because you start with one glucose molecule, you end up with 2-pyruvate. You are not adding pyruvate here all of a sudden to go back and make glucose through gluconeogenesis. You're using other non-carbohydrate reactant or other carbohydrate reactant depending. See how you can even begin here with glycerol. So it's all different. But it is called gluconeogenesis for the process of making extra glucose molecule within your liver cells. When there is an excess of glucose molecules that our body didn't burn up for energy, let's say we ate a big meal and then we end up being couch potato for a day or something, and now you have all this glucose floating around in your body that you're not using up immediately. So instead of getting rid of them with our waste materials through our feces, as we've discussed in the digestive system, it is our body's natural response to store them up just in case we need them later. And sometimes that just in case never happens and we just end up gaining weight. But this is just our natural way of how our body works. This is accomplished by the process called glycogenesis. So glycogenesis is where extra molecules of glucose are linked together in polymer chains called glycogens. So this process requires a unique molecule called UTP, or known as uridine triphosphate. I guess is another form or modified version of those adenosine triphosphate, guanine triphosphate that we were talking about. Okay, so let's say you then decide to go for a run on the treadmill and don't want to be couch potato all day long. So now you have depleted glucose from your bloodstream. So you need to replenish your blood with glucose again. So now you can go into your glycogen bank of glucose molecules, go through a process called glycogenolysis, which will break apart those chains with glycogen to release glucose back into your bloodstream. This is a very quick and simple and one enzymatic step process, at least compared with getting glucose from our fat storage, which we will discuss next. This glycogenolysis is a lot faster way to get glucose. So hence usually we go and grab from our glycogen bank reserve before we hit our fat molecules for glucose source or as a source of glucose. The tough part of this chapter is being clear about all these different metabolic processes names because they can sound very familiar. As you can see just from this slide alone, you have gluconeogenesis, glycogenesis, and glycogenolysis. They are not, differ by very much, and they sound very familiar. So that's why that chart from the beginning of the lecture is a very helpful one to make sure you are not confusing between different processes. So make sure to pause the video whenever you want to and go back to that diagram to ensure you're clear about each of these processes as we're talking about the macromolecules, catabolism, and anabolism processes. Okay, so let's have a break now and we will come back to chapter 25 part 2.